In this video, I will compare the analytical strategy and workflow of Magellan's Compass software to that of standard analysis methods. I will use a proteomics study for this comparison, but most of the differences apply as well to lipidomics or metabolomics studies. I will circle back to some specific points about these methods near the end of the video. Importantly, Compass works for any of these types of mass spec-based omics. For most proteomic studies, intact proteins are subjected to enzymatic digestion with trypsin or other enzymes to generate smaller peptides that are more tractable for instrumentation. Compass can handle mass spec data of these samples regardless of the proteolytic or other processing methods applied to the sample. During mass spectrometry, MS1 scans are periodically performed to catalog all the detectable molecules in the sample, along with their mass spec properties. Instrument software then selects some features detected in MS1 scanning for fragmentation and subsequent mass spectrometry, generating MS2 scans. The MS2 scans are used to establish molecular identity of the precursor ion features, peptides in the case of proteomics. MS1 scan data is seldom subjected to any analysis, so most mass spec software is not highly developed for the challenges of working with this data stream. In contrast, Compass analysis is focused on MS1 scan data. That means it considers the differential expression of every molecule detected by the mass spec, not the subset of molecules subjected to MS2 scanning. This eliminates bias introduced when the instrument software uses its algorithms to select specific MS1 features for MS2 scanning. Traditional analysis takes the peptide sequences derived from the MS2 scan analysis to identify the parent proteins and determine their abundance. Published reports refer to proteins and their abundance without reference to the underlying mass spec data. All of the expression comparisons and statistical validation is performed using protein-level data, meaning it is performed using a highly processed and curated subset of the data. Given that Compass uses MS1 data for its analysis, it works with unprocessed and non-curated data. One major advantage is that quality control problems with individual samples or groups of samples become readily apparent early in the analysis, allowing users to compare results that include and exclude such examples. Working with MS1 scan data also dramatically increases data scale because only a small percentage of the features detected in MS1 scanning are selected for MS2 scans. MS1 scan data contains information upon millions and millions of data points, whereas this is reduced to tens of thousands of data points in MS2 scans. In proteomics, MS2 data points, the peptides, are combined to generate data on thousands of proteins. Compass typically considers around 1 million data points per sample in its comparative analysis. This increases the sensitivity for detecting differences that are connected to the most subtle biology and gives far greater confidence when validating those differences. When performing LC-MS-MS with MS2 scanning, 90% of the instrument time is devoted to MS2 scans. Since Compass does not require MS2 scan data, it is possible to perform feature-first proteomics and collect only MS1 scan data by LC-MS. Without performing MS2 scans, instrument time required for each sample can be dramatically reduced even if higher resolution MS1 scan data is collected. A key difference when using COMPAS relates to the molecular identity of the data points being assessed. The sacrifice of data scale when performing MS2 scanning means investigators know the molecular identity of the data points they work with, though there is no guarantee the specific proteins identified during MS2 scanning have any connection to the biology under study. COMPAS focuses instead on the biological relevance of the data, using comparative analysis of MS1 scan data to find data points whose expression pattern is most closely connected to biology. This approach is sometimes referred to as feature first, as it puts the relevance of the feature ahead of its identity. An advantage of this approach is that it reveals whether biological signals exist and whether they appear to stand out above random noise early in the analysis rather than at the end of the study. It also is more likely to lead to the identification of unexpected or novel features. Where does molecular identity fit within the COMPAS analytical workflow? One answer is that it can direct molecular identity discovery efforts. COMPAS is designed to yield a list of MS1 precursor ions with strong connection to biology, and whose MS1 coordinates, meaning M over Z and chromatographic retention time, are known. This list can be supplied to the mass spec instrument for targeted identification, revealing their identity. When annotating features is technically challenging or costly, as it can be for certain mass spec applications, focusing those efforts on features with high connections to biology can be very advantageous. COMPAS can integrate molecular identity information into its analysis at any point, whether following a COMPAS-driven molecular identification campaign as described, or even at the outset of analysis. Existing MS2 scan data can be processed with standard software to generate a list of identities of MS1 precursor ions with their MS1 scan coordinates and identity information. This list can be supplied to COMPAS, which will then indicate when data points with strong connections to biology also have MS2 identity information. In turn, this can show what proportion of biologically relevant MS1 scan data has passed through to analysis in the form of MS2 analysis, as well as how much MS2 data is biologically relevant. Typically, this turns out to be a small percentage. In some instances, molecular identity is far less critical than the ability to determine sample classification based on the expression pattern within the mass spec data. Here, using COMPAS in a feature-first workflow is ideal. COMPAS is designed to use machine learning classification to identify data points with strong connections to biology. Trained classifiers emerging from this step can then be directed towards data sets from samples with unknown classification, predicting their classification with defined accuracy and confidence parameters. Metabolomics and lipidomics studies differ from proteomics studies primarily because the molecules under study are not biopolymers, strings of repeating chemical units. There are no standard catalytic reactions performed on the sample, as proteolytic cleavage is often performed for proteomics. Further, metabolites and lipids undergo fragmentation in less predictable patterns than for peptides, so MS2 scans are used to determine identity differently. None of this materially changes how COMPAS is used to analyze metabolomic or lipidomic data sets, so most of the prior points apply equally when performing metabolomic or lipidomic analysis. What kind of mass spec data will not work with Compass? Any files where multiple samples are subjected to mass spec in tandem, as with TMT labeling, will not work with Compass. Compass needs separate mass spec files for each sample. However, this point highlights another key difference of working with Compass, namely the ability to perform integrated analysis of multiple mass spec based omics types. When multiple distinct mass spec files, each from a different instrument run, derived from the same subject, standard comparative analysis is directed at batches of files performed with the same method. In other words, standard analysis performs parallel but separate analysis. Integration of the dataset occurs only once the mass spec data has been fully processed. In contrast, Compass combines mass spec datasets in such studies at the outset of its workflow for total and unbiased integration. Users can see the relative contribution of each mass spec method to the overall analysis, so users can make informed decisions about their value to their own research goals. We hope you will consider using Compass to advance your research agenda.